It's a great pleasure for me now to introduce to you Joe Felsenstein and Fred Bookstein, who will give a talk for us here today. So if you work in phylogenetics, you probably will know Joe Felsenstein very well. He is, I think it's fair to say, the father of modern statistical phylogenetics, and arguably maybe the father of the modern way we do phylogenetics today. This made numerous very, very important contributions, what we used to call seminal contributions. We can't call that anymore, but things that have seeded the field of phylogenetics. He was the first one that really showed how you could do likelihood calculations on phylogenetic trees, on DNA sequences, led to the modern way that we are doing phylogenetic inference now. He invented many of the ways that we use to quantify uncertainty about phylogenies. He was the first one to use bootstrap on phylogenies. He was also, even though it's fair to say also that he's not a big fan of Bayesian phylogenetics, the first one that actually did MCMC on trees as well, but for the purpose of doing population genetic inferences. And he has done and made a number of contributions to the comparative method and the way we think about comparing data from multiple species. Many of the ways we think about that today, we can really trace back much of the thinking to Joe Felsenstein. So it's a great honor to have him here today. He's inspired much of my own research through time. And I think anything that has to do with statistical phylogenetics will be complete by hearing from him. So it's very scary for us now that he is teamed up with Fred Bookstein, who is also very famous for being able to quantify statistically biological shape and the way shape transforms. So the same thing as we want to do in the center. So I'm thinking that the two of them together is sort of an explosive cocktail of what they might be figuring out in terms of solving this problem about how to model shape of phylogenies. And I believe we're going to hear about some unpublished, at least to my knowledge, research today that they've been doing recently. And the title of the talk is Insight into Old and New Methods, Geometric Morphometrics and Phylogenetic Inference from a Linear Maximum Likelihood Point of View. So welcome and thanks so much for being here, Joe and Fred.
I've given I'm sorry you're going to have to miss a full lecture by Fred Bookstein, and my lecture may be cut short a bit because of the time constraints and our computer problems from this end. A shorter title of the talk I'm going to give is Evo Geomorphometrics from a linear ML point of view. I'm not going to be showing you pictures of nice, I'm not going to show you 2D morphometric pictures because I ran out of time on the computational example. I'm very happy to be here in Copenhagen. I've been in Copenhagen before and enjoyed it very much. In order to have some of that experience, I picked up from a local bakery this, which I will eat after my talk, which is what's called a cheese Danish. Anyway, okay, that's the extent to which I'm in Copenhagen. Here I am in Seattle at 7:30 in the morning. Now, I'm going to give a rather technical talk, and I'm going to a little bit heavily algebraic, without really a computational example at the end because the programs involved are mostly not working. And what I want to, it's going to be joint with Fred Bookstein. And well, you've seen both of us already, but here we are, and including our common ancestor, JFL Bookstein, who we figure must have existed. I'm going to start out and talk about 2D morphometrics in this example. And I want to talk about landmark data and try to approach it not through the existing machinery of geometric morphometrics that Fred has contributed to so much, but to try to just see if I can make up a linear model or a nonlinear model to do the same thing. And when you do, one of the things you find out is that you have a form, you I don't know if my picture is, my own little window with my picture is big enough for you to see my hand gestures, but I hope it is.
Yes.
You find that the form has landmarks. They are XY coordinates. The form has a location in your space, whatever space that you're digitizing in. And that location is not a property of the organism, but is a property of the person who did the digitizing. And the way of getting around that is well known, is to do what are called Helmert transformations. And that's a whole machinery. But what it amounts to in the end is that the easiest way of dealing with it is to take your XY coordinates and get the centroid of the specimen, which is to say the mean of all the X's and the mean of all the Y's, and that gives you the centroid. And if you take all of your specimens and superpose their centroids, then that takes care of the issue of rotation. Let me see if I can. No, yeah, okay. But then there's the issue of rotation, because the person may have put the specimen down on the screen, on the digitizing surface at some angle, which is really a matter of, it's mostly a matter of them and not of the specimen. And it would be nice to have a transformation of some sort, a standardization of some sort that would take care of that automatically. And there is the generalized Procrustes analysis, which doesn't quite do that. But what I'm going to do is try to incorporate rotation into the model. And really there are three different ways. One of them would be to use a generalized Procrustes system and just superpose all your specimens, rotating them. I'm going to use not a Procrustes, but what Fred and I have come to call the Boas superposition, in which we don't rescale the sizes of the specimens. We superpose their centroids, and you get rid of the rotation with Procrustes-like machinery, but with the part left out that changes the sizes, because size is a real biological property. And you do want to do analyses that talk about things like allometry. So you want to leave size in from the beginning. But you can also make a statistical model of rotation. You could say, well, suppose there are independent small rotations of each specimen, drawn from a distribution. Perhaps the angle could be drawn from a normal distribution with a small variance. You can also incorporate into the machinery, allowing that rotation to co-vary with features of the form. So that leaves the interesting possibility that if a form, for example, grows a horn, has a dramatic change in shape, that might affect through its effect on the psychology of the manual steps or whatever automatic steps you have. It might affect the angle, and you can actually put that into the model. So that's going to be the way we're going to deal with rotation. And when you start doing a model, excuse me, you start doing a model of forms that are have XY coordinates. Here's a vector of a form, x1, y1, x2, y2, all the way up to xp, yp. And if we make ourselves up a model, not necessarily a linear model, here's what I get, which is that the coordinates of the form are changing, and the model of their change is going to be that at time t, suppose we're at time t and we're going to time t plus a little bit, t plus delta t. we're going to have the form, and adding on to that, we're going to have some random variables, which are covarying, and times a delta t. So we're imagining a small move as a form evolves up a lineage, and we're asking about the change in a small amount of branch length delta t, and we're going to, for the moment, not say that that's Brownian motion. It's something like Brownian motion. And we're also going to be talking, separating size and shape. Remember that we kept size in our analysis. So we can say, well, here are the changes in shape. And over here is a term for size. Suppose there is a size random variable for the change in size. And it's going to multiply the current form, basically moving everybody out or in from the centroid by an amount which is also a random variable. We'll come to rotation in a moment. This is just a form evolving up a tree on a single branch or a single path all the way up to a tip. We're leaving out the rest of the tree for the moment. Well, here's a model, and you look at it and say, okay, you've got a statistical model. What kind of a model is that? And the answer is it's not a linear model. And it's not a linear model in this case because of one important reason. The size change depends on the current size, which is a random quantity. And it depends on a random variable, which is the wandering of a size quantity, theta. And so the product of these two random variables means that this isn't linear. Now, how do we get around that? The usual way, and at the end, sorry, after you go with this process up to the tip, then of course you have to think about the rotation of the specimen that occurred up there at the time it was digitized. And here's the rotation matrix. Let me see, my mouse is also not cooperating. Here's a rotation matrix, and basically what it just says is there's some kind of rotation. It's a random variable also. And here's a little 2x2 matrix. That's the rotation of the first two coordinates, the x, y for the first landmark. And here's one for the rotation by the same amount for the second landmark, all the way down through the landmark. So they're all getting rotated. The vectors that are coming out from the centroids are all rotating by a little bit. That again contributes to the nonlinearity of the model, because the rotations there's a random variable, and then there's sines and cosines coming in. And it's the change due to rotation also depends on the current form. So it's a product of some cosines and sines of a random variable theta times the random quantities z or z. That's the rotation part. So we have a nonlinear model here. Now, to make it linear, what's usually done is to assume that the one way anyway, is to replace the z in those terms. Let me back up for a second. Replace the one over here and the z's in the rotation term by an average form, which we can take as the starting value on the lineage. So if we replace those z vectors by the starting values, we'll be close in terms of the changes of size, and we'll be close in terms of the rotation. And then you have a linear model. So if I now look at it and say, okay, what have we achieved by doing that? The answer is, well, basically, we're doing the tangent plane approximation that's familiar to geometric morphometrics for the changes of shape. We are keeping size in the analysis. We're putting it in an approximate way. So we're getting something that's very close to ordinary morphometric practice, except that we're keeping, in this case, keeping rotation in the model. Now there's the tree. If we have the linear model, we can we've been talking about the changes of a single lineage, but there is the whole tree. If you have a linear model, you can do the machinery that people have been starting to do in morphometrics. I have to say that morphometrics has been slow about taking into account phylogenies. But in recent years, in the last couple of decades, there's been a gradually increasing interest in that. And I take it that that the center that whose symposium this is going to do a lot of that. Now, if everything is multivariate normal, which it is in the linear approximation, then you can take the tree, you can do things like contrasts on the tree, and the machinery basically works out. You can basically estimate the covariance matrix of changes under the Brownian motion in this size knowledgeable version. You can also you can also estimate parameters for size, the rate of change of size, and you can estimate allometry. So let me move along. Pushing the tree aside for the moment, we can estimate the parameters of the linear model by using multivariate normal machinery. I'm not going to go into the details except to say it's not a trivial matter if you use maximum likelihood. And you say, okay, we've got a linear model. Let's estimate the parameters by maximum likelihood. You want to estimate the starting form, and you want to estimate the shape parameters and the size parameters. And if you look at the way that that's set up, I just want to say that it isn't completely obvious how to do it. It's not simply a matter of using the empirical covariances. Let me then talk about, okay, you got this linear model. It's something like standard morphometrics. What are you going to estimate? Now here, Fred has very strong opinions about this. I have much agreement with his views, but I think often we're forced away from having a very explicit model of the change of the shapes onto doing a more empirical model, such as using principal components and to estimate important directions of the variation that we see empirically when we follow landmark data up a tree. And if we have some actual mechanistic model, for example, or understanding of the Evo-Devo machinery of that form, we could do better than just doing a principal components analysis. And we would then have something that I'm calling principal components with a PLE instead of a PAL. We'd have a principled analysis of the form. Fred will be able to tell us more about that. It's been an important emphasis of recent writings that he has. And now I'm going to say something completely heretical. If you've got a linear model and you've got your forms and you've either removed the rotations or incorporated them into the model, what about the morphometric consensus? Well, the morphometric consensus, as we know, is just to use things like generalized Procrustes analysis. Here, let's imagine that we have in a high dimensional space our forms. So each of these points is a whole form with, say, 20 coordinates of 10 landmarks, for example. So here there are standard morphometric consensus methods, as I understand them, is to get distances among these points. Then you go through the principal coordinates machinery and you get morphometric axes, and you're now in a morphospace. In that morphospace, one of the things you might be interested, if you're not able to do the more mechanistic stuff that Fred is talking about, is things like principal components. So here's PC1. And there is machinery in the standard morphometrics for then going back and looking at where all this is on your original forms. But I just want to make the point that the analysis that you will have done going the route of the morphometric consensus ends up being exactly equivalent to taking principal components on the original high dimensional points and just simply ignoring most of the morphometric consensus. So we're going to take a rather heretical position and say, do we need to work in a morphospace? Answer, no. The standard morphometric consensus in the end is just another way of getting the same results. And the danger is going to be that we may think we've done something special by going off to morphospace, but in the end, by going off and coming back, we end up where we started, or we end up where we would be if we just stayed in our original superposition of forms and estimated, say, principal components there in a phylogeny-aware method. I'm leaving out here the phylogeny steps, but they're there in both routes through the diagram. Okay, so that's our second heretical thing to say. I think, I think so far, third heretical thing to say. First, leave size in. Second, watch out. Principal components are not necessarily the best way to go if you can have any kind of model of what the forms are doing. And the third way is morphospace. Well, we don't use it. Now, suppose we consider the nonlinearity of the actual model and don't make the approximations that I talked about, which end up close to standard morphometrics. Well, one of the things you could do would be to do a Markov chain Monte Carlo analysis. Now, I take it that that's going to be a matter of considerable concern already to the people in this workshop. So perhaps this isn't a very novel statement, but what I think I would envision in an analysis that I don't yet know all the details of is to say that you could take a tree obtained, say, from molecular data. You could use the nonlinear model that I started talking about before I started talking about approximations. And you could estimate forms at various points of the tree. In fact, you could divide each branch into a number of segments. And then you could do Markov chain Monte Carlo sampling of what those ancestral forms were. You know the forms up the tips. And that will involve things like Gibbs sampling or perhaps Metropolis sampling. So you may be updating a point like this one that I'm pointing to on the tree. Here we have a picture of the three immediate neighbors, and we update this red point in view of the three neighbors. Now, if we had a linear model, that's very easy to do. If we have the nonlinear model, it's a little harder because we're going to have to imagine changes that respond to the forms nearby, thing nonlinear. And we could update interior points that have 2 neighbors or interior points that have 3 neighbors. We could imagine doing a more complete nonlinear model that takes into account the way size comes in and takes into account in the end also rotational terms, and doing it by MCMC. I'm not going to show you an example because we don't have it working. Maybe one of you does. But finally, okay, if we want to use nonlinear models and not approximate them as linear, and we want to do them in all the ways we talked about, is that really good enough? And I think the answer has to be that as much as all this kind of machinery solves the geometric problems, it still leaves us making rather arbitrary assumptions about our random changes coming from multivariate normal distributions, even when you take the nonlinear terms. And even though we can do the Markov chain, we can imagine doing the Markov chain machinery, it still leaves us without knowing about or using the developmental processes that produced these forms. If we could know something about the genetic variation and which directions, which linear and nonlinear ways it works out. And if we can know about things like the directions in the space that selection pressures are acting across the tree, we would be able to do a much better analysis. So what I want to do, just finishing off, is dream about a future morphometrics that is not only geometric morphometrics, which has been going on up until now. It's not even the Evo geomorphometrics, which is basically, I think, what a lot of people will be talking about at this very interesting workshop. But in the long run, what we need to do is somehow integrate that with a knowledge of the development, the Evo-Devo, the developmental biology underlying these forms. Now, that's easy to say. What I'm really doing here is kind of cheating, because I'm saying, imagine that you could do it all with an informed knowledge of the developmental processes and the ways in which genetic variation is or is not available. That would be an Evo-Devo geomorphometrics. But I think it's always helpful to keep long-term goals like that in mind, because it helps you avoid getting stuck concentrating on very limited methods when you can look ahead and see that really this is part of a bigger effort. Ideally, we could think about an Evo-Devo geomorphometrics that interacted with our knowledge of developmental biology and our knowledge of the natural selection on these forms, such that we could have statistical information provided to the people studying development, and we could make use of their experimental and empirical results. So there's a dream of a future Evo-Devo geomorphometrics. So therefore, as we go forward, there will be decades of work on this, and I can claim entire credit for all of it just by having described it in vague generality. So I have a few references here. People probably know about my work on phylogenetic contrast. There's also a paper I published in American Naturalist in 2008 on how you take within species variation into account. And I have another paper on discrete traits and how you can correlate them with continuous traits. And that's an American Naturalist in 2012. It's an MCMC method. What I'm hoping to do is take that MCMC method and implement some of the nonlinear analysis that I've been referring to here in those programs by expanding them, and also take within species phenotypic variation into account. Fred sent me a lot of references, but I ran out of time to put them in. He has many papers recently about making some of the points I've just made. Maybe Fred would like to make a few comments now, because I've run out of things to say.
First,
Back
I'd
to
like
you all,
to say that,
particularly Fred.
yeah, what I do isn't really Jeremiah ads. I'm going to start with a very small advertisement for my textbook here from 2018, which hints at most of what Joe was saying about the role of rotation and size, without talking much about the phylogeny part. But there's one example here near the end in which ignoring nonlinearity really changes the answer that you come up with. And that makes it important to, as Joe was saying, recenter these analyses. I'd like to take my 5 minutes here to connect to the previous talk, which talked about the structure of a specimen record in Morphosource, and in particular, this concept of metadata. That concept has been developed quite a bit more highly in other branches of applied biometrics. In particular, there's a version that starts with anatomy, something called the foundational model of anatomy, which exists in enormous detail, 30,000 entries for humans, because it's used in medical education, but should exist also for extensions, at least to the family of mammalian skulls, primate skulls, larger scale structures of vertebrates. Richard Raymond for decades was working on conceptually on something equivalent for his microfossils. And the idea of these models is what is usually called an ontology, which means a computer readable linkage between the terms that are used and other kinds of literature. So I'm on a paper that has an ontology of physics in biology, what you would want to know about a measure of, say, the diameter of an aorta that has something to do with the function of the aorta. And in the case of mammalian skull data, like those examples in the previous talk, this would be an analysis that says some of these landmarks are on one bone, some are on another bone. Those bones have names and homologs, but also in terms of connections with function, connections with development, some of the landmarks are around a cavity, the gape, which Melvin Moss called memorably the functional matrix of these ideas. And to make sense of a particular collection of skulls, not a single example, but a collection, you would want the metadata somehow to be extended to include these aspects of the ways that explanations are brought to bear on the description of their variability. To repeat, this is now going on in the medical literature. Publications about chemicals, about molecules, are accompanied by links to the metadata of what that molecule is supposed to relate to. So you know what other articles to bring up. You can think of that as what Joe was talking about as the incorporation of either development or selection, this evo-devo part, into the analysis of whatever sample of forms you got. The metadata is going to be used to decide how to collect multiple specimens. And the people who are doing medical literature surveys know that this is essential because you need the computer to help you to do that, because there are so many articles, as much as you need the computer to help you to do that in museum collections because there are so many forms. I'm guessing there are millions of beetles and millions of butterflies, and you need to know which landmarks correspond between species when they do, which parts of the butterfly match explanations of the aerodynamics and so on. I'm improvising here in part because my time is limited. And so I don't have a display of the references, but that name, computed ontology, or ontology of physics in biology in this case, should be considered as a part of this discussion of metadata. The metadata doesn't pertain to the specific landmark. It pertains in effect to the literature that refers to the name of that landmark across species or across developmental stages. And then to conclude the comment, the Procrustes method of standard morphometrics totally ignores this. Procrustes method obliterates all of this metadata and accounts for much of the frustration of people like me and my colleagues in establishing connections between morphometrics, biometrics, multivariate analysis, and all of the rest of the things that biologists like to do, which at the very highest level is to turn arithmetic into understanding. So metadata is ontology. And with that, I'll hand this back to you, Joe, or to the session director for questions.
Well, thanks so much to Fred and Joe. Oh, did you want to say something, Joe? Sorry.
I just want to say that I will assume that having heard about this from me and Fred, you will all now completely change the ways you do things.
So are there questions either from people online or in the room to Joe and or Fred? No questions from anybody? We're all convinced we're gonna do morphometrics, phylogenetic morphometrics, completely different from now on. I'm on board. You sold it to me, Joe. Well, let's thank Fred and Joe again. Oh, they've got a question.
I could make a comment on I really appreciate Fred's point about ontology, and I think there is a way to establish rich ontologies on Morphosource, but that is essentially something that has to be grown by the user communities because you can't source it from the museum directly. You know, I would say the best developed ontology for anatomical terms that I'm aware of is called Uberon. And so in the coming, I would say, year or so, we do want to do essentially a matching protocol and re-index our anatomical part descriptions according to the Uberon ontology. I think there are other ways, actually through morphometrics, with standardized ontology for morphometric landmark terms, that we can essentially enrich the data that in online resources even more along the lines of what Fred was mentioning. So I agree.
Yeah, thank you. That is also what happened in the foundational knowledge of anatomy literature, that the structure of this archive was established basically by one person, Cornelius Ross, here in Seattle, and then thrown open to the whole world of people basically using computers to help teach anatomy. And the user input ended up dominating the structure of the final release. it was very important to have a way of opening it up to users that started with something and then got better. Maybe geometric morphometrics is like that. It sort of started with me, and then it gets better by the input of others, younger with blacker beards, in the case of the males. Thank you for that comment.
Any more questions? Online, you can just unmute yourself. Or comments? All right, let's thank Joe and Fred again.